Okay, so I will do a general introduction to BioAster and artificial intelligence in the role of drug discovery. And I'll pass over to Lucia, who will go into more detail about the power of artificial intelligence, machine learning, and then finally Sophie, who will give an example of some AI-powered technology developed at BioAster as an internal innovation. So, my name's Derry Mercer. I'm the head of the antimicrobials program here at BioAster. I've worked in antimicrobial drug discovery for the last 20 years, and recently I got a much greater interest in artificial intelligence and deep learning capabilities for the purposes of drug discovery. So BioAster is a technology research institute based in France. It's a unique public-private partnership, not-for-profit organization who facilitates research into microbiology and infectious disease. We do this by helping our partners design and develop novel antimicrobials in the case of the antimicrobials program. We do this through scientific expertise, available technologies, cutting-edge technologies, and also in-kind contributions to help people with their research problems. There are 4 fields of applied microbiology at BioAster's, the antimicrobials program, which I head, the vaccines program, the microbiota program. Vaccines program works predominantly to understand the mechanism of action of vaccines and vaccine adjuvants. The microbiota program, and then also that we have a diagnostics program to develop sensitive and cost-effective solutions for diagnosis of infectious disease. The microbiota program has the ability to cultivate organisms of interest from complex microbiota and to study host microbiota interactions, including phenotypic characterization of these organisms. We're able to do this through our technical units. We have a clinical microbiology and a molecular engineering technical unit who do all of our microbiology, including both in vivo and in vitro, where we can do BSL-2 and BSL-3 experiments. We have a preclinical models and imaging unit, bioassays, microsystems and optical engineering unit, which does a lot of our immunology and cellular and molecular analyses to generate functional assays for use in microbiology and infectious disease. We also have an omics hub where we can do all sorts of omics, genomics, transcriptomics, proteomics, metabolomics, and also all the relevant and associated bioinformatics studies. And then finally, we have a data management and a data science technical unit, which do biostatistics, but also cloud computing, artificial intelligence, and machine learning. This means that we can cover all of the preclinical and some of the clinical phases of the antimicrobial drug discovery program, from novel screening methods of efficacy and toxicity, looking at mechanism of action, dose optimization, resistance determination, looking at single or drug combinations and the host responses to infection and therapy. So, artificial intelligence and machine learning, what are they? Artificial intelligence is a technology that enables computers and machines to simulate and solve problems. Whereas machine learning and deep learning algorithms use neural networks to process large amounts of data. The classical machine learning algorithms use neural networks with an input layer, 1 or 2 hidden layers, and then an output layer, whereas deep learning algorithms use deep neural networks, which have an input layer and output layer, but often 3 or more, sometimes hundreds of hidden layers. And these multiple layers enable unsupervised learning. They can automate the extraction of features from large, unlabeled, and unstructured data sets. So, are artificial and deep intelligence and machine learning the future? Not really, no. They're already here and have been for quite a while. Online shopping, advertising, web searches, digital personal assistants and chatbots. language translation software, self-driving vehicles, and cybersecurity are just a few examples where these are important features. It's estimated now that 37% of organizations have implemented AI in some form. And it's predicted that by 2025, 95% of customer interactions will be powered by AI. Even dating apps are now using AI. It's becoming that widespread. So how can we use them in science? We can use them for improved disease diagnosis. For example, the AI algorithms can be trained on much larger data sets than a radiologist for diagnosis of cancer and other diseases. They can be used to monitor health status, people wearing blood glucose monitors. That data can be transmitted directly to healthcare providers and analysed and recommendations made. It can make the use of electronic health records smarter, faster, and more efficient, whilst reducing costs associated with them, and also makes the analysis of healthcare records, in which there are huge amounts of data, much more efficient. They can be used for data analysis. For example, multiomics generates huge amounts of data that AI can process much faster and much more efficiently than humans possibly can. Even virtual health assistants, which can simulate human conversations to offer personalized patient care based on patient input. And of what we're most interested in, drug discovery and development, this can be for target identification and virtual screening, chemoinformatics, drug interactions, formulation and delivery, and also also biomarker discovery, pharmacovigilance, including assessment of adverse events for clinical trial patients not based on site. So I did a recent PubMed search between 2010 and 2024, and this generated almost 5,000 hits, of which some of the more impressive ones are listed here. I don't know if you've ever noticed the best match function in PubMed is actually based on AI. So I'll now pass you over to Lucia to give you a bit more information on the BioAster's machine learning and AI capabilities.
Hi, guys. So I would like to start these few minutes of conversation by a bit introducing myself because it would give a little bit of a context. So I am now the head of a team that is doing data science, machine learning, artificial intelligence, deep learning, among other things. And I have a background in biology, healthcare. I have a master's degree in pharmaceutical biotechnology and a PhD in neurobiology. So, I'm a big passionate of biology and everything healthcare. However, I discovered about machine learning and really fell in love and became I'm now a strong believer in the capabilities of deep learning in healthcare. So I am totally biased, and you will see the passion in my eyes when I'm going to talk about the slides that I prepared for you guys today. First, I would like to introduce a little bit what machine learning and deep learning is and what it can do. So we can say that artificial intelligence is a very versatile tool. We can analyze a lot of data with it, data that range from, graph, like networks, for example, protein-protein interaction or protein-other structures interaction, structural data. In this case, I presented here Raman spectra, image analysis, but also text or anything that is related to a time series. So for example, if you have to analyze different time points, anything in which the time element is relevant. And there are different frameworks that we can use to infer information and business intelligence out of this data. If we have a specific outcome that we want to predict, then we are going to use supervised learning. There are a couple of examples here of things that we can do with supervised learning. in one case we want to predict like a class. Is it better to give the red pill or a blue pill? Or maybe we want to predict something that is trending in time, then we do like forecasting. For example, how bacteria are growing. If we don't have a specific label we want to predict, but we want to just to extract information from the data, then we are going to do some sort of classification clustering of the data. And in this case, I presented here an example of hierarchical clustering of bacteria's taxonomy. I would like also to present some of the things that changed in the field of machine learning and artificial intelligence that can be, and how they are extremely interesting in the field of antimicrobials and drug discovery and drug development. And one of these concepts is really embedding. So an embedding is the transformation of input data into a string of numbers of a set dimension. And then we put these numbers that ideally have distilled all the information of the input inside a space that is called an embedding space. That is really semantic, it has a meaning. And this space has some properties so that similar things are really close by in the space, and very distant things are semantically very different. And there are also some operations that we can do in this space. In here, I present some examples of words, but you can as well translate this into the field of your interest. So the same conversion that we see from Paris to France, we can see from London to England. And so the way we can use these embeddings in terms of antimicrobial is, for example, to find similar molecules or similar structures, or maybe find a different drug target, or maybe find a different proteins that from similar species, but that are unique for that specific species. Now, I said that we can analyze a lot of different kinds of data. And the way this data can be analyzed is through specific frameworks that were developed in the years. And one thing that I noticed between the difference between the biology field and artificial intelligence is that in machine learning, things are proceeding really, really, really fast. The things that in the past were considered extremely complex and very difficult to analyze nowadays are a reality, and they are, very common use. So we can analyze the networks, so these like protein-protein interactions, for example, with a specific framework that is called the graph neural network. We can analyze tabular data with, like a feed-forward dense neural network, where there are different layers, as Terry was mentioning before, that go from the input to the output. And in the middle, there are a number of hidden layers that would transform the input data and would be able to generate, to learn from the data to actually predict the output. From images, we typically use a framework that is called the convolutional neural network, in which the image is divided into different patches, and there are other transformations that the model learns from the training data set to go from the input to the output. And regarding anything that has a sequence like text or maybe, amino acid sequence, there is another framework that is called the recurrent neural network, in which the model really learns the time dependencies between the different inputs. But nowadays, we also have other frameworks that are super interesting that have been developed a lot lately, that are generative models in which you have an output that is generated by itself. And here are examples on the different architectures of these models. And you can see how all the different deep learning methods are very different. So now I'm going to present you some examples in which these frameworks were used. In this specific case, the authors of the papers were analyzing, were trying to understand better the different components of the inputs for a clinical case in which they were facing a resistant species of Staphylococcus aureus, and they were given time varying, of course, antibiotic treatments, and you can see them marked with the asterisk. The output here is the Y, and it is the patient survival. And there are a number of inputs, and this framework that I'm going to describe you now is really capable of distinguishing the influencers, which are the inputs that influence the outputs, which are the confounders, and make a distinction between what is causative of the outcome and what is not. And so we can use these kind of frameworks to distinguish all the different components of these inputs, but also to optimize a certain situation. For example, we can optimize the antimicrobial resistance onset, for example, if we can understand what are the inputs that are correlated with the output, so the antimicrobial resistance, and which are causative. And so in this way, we can really improve the situation. Something that really changed the biological field in terms of artificial intelligence is AlphaFold. That is a framework that in the top panel. There are a lot of different blocks, but this model gave extremely good performances to predict the 3D structure of proteins from simply the input of this protein. And you can imagine how this was important for a lot of downstreams applications in which the 3D structure of a protein is probably the limiting factor. There are nowadays also some models that are doing a similar thing for RNA, and so hopefully very soon we can cover all the different macromolecules. And these things can be extremely important, for example, when we are studying docking or designing a new drug, or maybe even doing drug repurposing optimization. And here is an example of how AlphaFold is used and the capabilities really of deep learning. In this paper, the authors were able to design a model that could really learn the sequence of the protein in such a deep way that the model was able to generate text for the protein's function. And you can understand that understanding the text of a protein just from the primary structure is really, really complex, and the results in these papers are really good. So we can really use machine learning for specifically optimized tasks that can be also semantically very, very different and complex. Just to summarize, this is a work that was done in which the whole pipeline was done in silico. Specifically, they were using a database for protein in which they were using different steps for in silico prediction of antimicrobial peptide prediction, and in silico docking, and then a molecular dynamic simulation. So all these steps are now possible in silico, and this is really a big time and money saver. So a few take-home messages. The machine learning field is proceeding at a really fast pace, and so whatever was unthinkable until a few years back now is reality, and this gives hope that the future is even more brighter. And in silico approaches can really speed up the antimicrobial development process and efficiency reducing the cost of experimental studies, because now we are not focusing anymore on hundreds or thousands of molecules, but only on the handful that were predicted being positive. And then AI can also be a good way to analyze the data in a new way. And an example of this is HoloMoa that my colleague Sophie now is going to present. Thank you.
Hello. So I'm Sophie Disneuf. I've been working in BioAster for 10 years now. I'm the lead of all the optical activities in BioAster. I have a background PhD in physics, and I've been working in optics-based diagnostic methods and also methods around the life science and antimicrobial discovery based on optics. So I will present the HoloMoa technology now, which is an internal innovation at BioAster. Well, the context, you'll know it, there's an increase of antimicrobial resistance and a crucial need to develop new antimicrobials, possibly with new mechanisms of action to guarantee there is no existing resistance in nature. But the reference methods of early stage high-level mode of action detection and mode of action novelty assessments are labor intensive and costly. And so the pharma drug developers in general express the need of a technology able to perform alternative phenotypic screening of drugs, which would be simple, fast, low cost, able to treat complex therapeutic solutions, and able to detect novelty of a mechanism of action. The game changers we thought of are the family of is computational optics and the family of quantitative phase imaging, which can be applied in a very simple design like the digital inline holographic microscopy I will introduce, which enables to obtain very informative images in a label-free manner. It's robust. Since it's label-free, we can do time-lapse and take advantage of the information contained in the time. And so it's called quantitative phase imaging, as we reconstruct not only the morphology of the bacteria cell, but also the quantitative phase. I'll explain it later. And of course, the second game changer is artificial intelligence and deep learning in particular, which is well known to classify very well images at the cost of having a very large database, to build very large databases. So the idea of HoloMoa is to develop a screening tool of molecules based on a combination of digital inline holographic microscopy and deep learning. And we did a demonstration in 2 steps. First, to classify molecules according to their mode of action when they had a known mode of action. And second time, we demonstrate that we could do detect the novelty of the mode of action of a new molecule. So here's just to show you an example of a traditional method for high-level MOS screening, which is called macromolecular synthesis, MMS. So it is based on assays to classify antimicrobial molecules into 5 inhibited biosynthesis pathways: cell wall synthesis, DNA replication, RNA synthesis, protein synthesis, and fatty acid biosynthesis. It has several drawbacks. The main one is that it is based on the incorporation of radiolabeled precursors into macromolecules of each biosynthesis. So no one really wants to do that anymore, I suppose. It is done by CROs. It is quite costly. And on the technical note, it has some drawbacks, such like rapid killers all lead to 5 positive assays, although they might have different MOAs at the start. Also, these assays cannot distinguish inhibitors effective different steps of a same pathway, and it cannot identify novel they cannot identify a novel mode of actions. And it's also quite slow to run this assay. So the idea to use microscopies to screen molecules and determine mode of action is not new, of course. One could use a transmission electronic microscopy, for instance, but it's painful and difficult, and not everybody can do it. About 10 years ago, started appearing some optical microscopy-based methods. I'm showing a few examples here. They are based on on fluorescence labeling, sorry. On the left-hand side here, you have, for instance, the bacteriological cytological profiling method. So relying on mainly two staining of different elements of the bacteria cells, the cell wall and the DNA typically. It was first implemented here with confocal microscopy. Since then, other developments used more common microscopes, epifluorescence microscopes, which give less resolution, but still, when you they also introduced machine learning to classify these images containing up to 4 labels, I think. But the main drawback here is that you still have the fluorescence labeling involved, and also since DAPI staining is involved, which is cytotoxic, you only get an endpoint measurement, and you cannot exploit the time dimension of the incubation with the antibiotic. So that's how came the idea of using another optical microscopy. Quantitative imaging, quantitative phase imaging technology. And we choose to use the simplest configuration possible, called digital inline holographic microscopy. It is very a robust setup, low cost, a simplified microscope using only a LED, not even a laser. The sample, you have here bacteria captured on a cover slip. And the principle is that the light, which is going to be scattered by each bacteria, is going to interfere with the light which passes without being scattered, without being disturbed by the sample. And you record an hologram, which looks like this, where you can see interferences patterns around each bacteria. This is acquired out of focus on purpose to be able to see this interference pattern. And from this simple experiment, there is some data processing after that, of course. We reconstruct, we do a digital refocalization, which is also called a reconstruction, and reconstruct a phase map, a quantitative phase map of the field of view. You can just remember that the phase is a quantitative representation of the thickness times the refractive index of the object. So this method is, say it again, label-free, phase quantitative. The different colors you see on the objects are the redder and the more highest the phase of the object. It's non-destructive. We can run time-lapse experiments and have a single cell resolution. And it's quite versatile because we could study any type of, I would say, any type of cell and any type of treatment as well. So the our HoloHemoA technology relies on this pipeline. We built a prototype able to run 4 conditions simultaneously. So the bacteria are first captured on a glass cover slip, which is used to close a small chamber, only 25 microliter in this case, where we introduce the growth medium and the antibiotic. We could use several concentrations of the same antibiotic or different antibiotics. We record time-lapse holograms, so every 42 hours, every 3 minutes. From these holograms, out of focus holograms, we reconstruct 40 phase maps, sorry, for each time, for 40 time points. And then with some registration and segmentation, we obtain dynamical phase map patches, phase patches, sorry, like this for each single bacteria. And this is what is used to feed the artificial intelligence pipeline then. First, for the purpose of MOA classification, if the MOA is already an existing one, and second, in the novel MOA detection pipeline. This is just a little side experiment just to compare the digital inline holographic microscopy and the fluorescence microscopy with the common DAPI staining. So you might see maybe that the for some antimicrobials, here I'm just showing two. We could see something resembling between the DNA staining here and the phase hotspot we see here. And most of all, once again with the DAPI, it's an endpoint measurement. While with our technology, we can follow the time, which we believe is very informative. A few examples here of the database we acquired. So it's just an example of one molecule in each class of mode of action: cell wall, DNA, RNA, cell membrane, and protein. And you have the untreated control here, where you see the cell dividing. For the cell wall, for this molecule, you see here an elongation and a bulging appearing. The phase becomes very intense at the end. Here some elongation, and maybe you see that here the three last molecules here tend to freeze the bacteria. So they are quite actually resembling on the morphological point of view. But we believe that the quantitative phase we measure inside the object and the fact that we have the time could help, and the use of course, artificial intelligence could help distinguish these families still. So the first goal was to predict the class of each antimicrobial we tested among a set of predefined MOA classes. I'm not going to enter into the detail, just you know that convolutional neural networks are commonly used now for extracting image features, normal 2D images. Here we have a third dimension. We have the time, which can be treated in with different options. We can use it at the same level, I would say, as the space dimensions, and use what is called a 3D convolutional neural network. Or a recurrent neural network can also be used as specifically designed for treating time images. And Lucia introduced them earlier. Okay, I'll just jump to the results of the classification. So first, I present the entire data set we acquired. So we have 22 molecules in total. Some classes like cell wall are much more representative than others, like cell membrane, for instance. And we have about 1,700 single bacteria patches in our data set. They were recorded for the one wild type strain, Escherichia coli, ATCC 25922. And so it's not an hyper-susceptible strain, it's a wild type strain. And you get the confusion matrix on the right here. So 80% of the data set was used to train the neural network architecture, and 20% of the data set was used as a validation, as a test. And so you have the prediction for this 20% here. compared to the true label here. And you see that we obtain quite a good diagonal with a sample level accuracy at 89%. We can note that there are still remaining issues, in particular the confusion between protein and RNA classes at some level, which we believe can be imputed to low statistics for some antibiotics. And maybe here the lack of molecules as well in this family. There were no existing molecules for gram-negative bacteria in this family other than pholistine. So we believe that if by increasing the statistics, we would increase the performance of the classification. So the second demonstration, the main purpose of the tool actually, is to detect a molecule with a new mode of action, be able to say that this molecule presents a new mode of action. Here again, there could be several deep learning approaches. We've chosen to use Siamese networks, which were inspired by face recognition problems, where you use two neural network, convolutional neural network, and pairs of patches in entry, and classify them as similar or different. So once again, we used a part of the data set as most of the data set as training, part of the data set of the non-MOA as test, and we used as well a new molecule with a new mode of action in the test set as well. So this new class, it's written already here. This new class is actually not new. It's a folic acid synthesis inhibitor, but it is new for our data set. It was not introduced at the beginning. And we didn't use it to train the algorithm. We just use it to test. So the molecule we used is the trimethoprim sulfamethoxazole mixture. So the Siamese network architecture computes at the end a similarity score. And if the score is close to 0, means the two series of patches are similar. If it's close to 1, means they are different. So you have here this coefficient, similarity coefficient, represented here for the test set compared to the training set. And you have in the first column the new molecule, which is not anywhere in the training set. You see that we obtain quite a high score for this molecule, while for the other, we obtain a high score, a low score, only when comparing with its own class. So we show here that this model recognizes well the non-MOAs and detects the new molecule as a novel MOA. It's represented here a bit simpler when we just added a threshold, and you can see that all the first column is orange, meaning it's a novel detection. So as a summary, we've developed two time-lapse label-free holographic microscopy plus deep learning pipelines. We obtain 89% MOA classification accuracy at sample level and encouraging novelty detection demonstration. As the perspective, we really believe that we would obtain performance improvement with a bigger data set, in particular with increased statistics. So we would like to work on that. And of course, we would love to test real new drug candidates with potentially novel mode of action. Then we would like to extend the demonstration to other pathogens, escape, fungi, and parasites. As a parallel work, so work in progress, we are developing now the RAMOA technology based on Raman spectroscopy and as a complementary technology to holographic microscopy for MOA detection and novelty assessment. So just in a few words, so Raman scattering is used to probe the Raman scattering spectroscopy, sorry, is used to probe the vibration modes of the molecules within the sample, within the bacteria. We have a optimized Raman microspectroscopy instrument in BioAster. So you can target individual bacteria and measure very quickly and efficiently the Raman spectrum for a bacteria. And what is a Raman spectrum? Well, it's a chemical fingerprint of the treated bacterium. So these peaks are due to the vibrational mode, are a picture of the vibrational mode of the molecules in presence. We can do some, first of all, of course, we can do some biological interpretation, comparing which peaks evolve and in which direction for the different mode of action. And of course, using machine learning again with this data, we can do MOA classification and novel MOA detection. And finally, I want to thank all the people involved in these two projects. The optical team, Zoé Sodaga did most of the work, and Elodie Bautrel as an intern. For the data science, the artificial intelligence part, Benoît Courbon did most of the work, as well as an intern, Darius Abedini, and Alexei Novoloaka as well for some statistics study. The microbiologist experts, Pavel, Laetitia, and Hélène, Melanie, and Crystal, and of course, some people for the data management as well, because we have to deal with large data sets, quite heavy data sets. And I wrote here as well, I'm not going to cite all the names, sorry, the leaders of these people around here. So thank you very much, and we welcome questions, I think, now.
So just to conclude, the artificial intelligence and machine learning fields are proceeding very rapidly. And we're firm believers that in silico approaches could accelerate antimicrobial R&D and improve efficiency while reducing costs. And to that end, BioAster has made significant investment in its artificial intelligence and machine learning capacities over the last 12 to 24 months. We believe that AI can provide alternative ways to analyze data sets, and they have been used successfully to identify the mechanism of action of antibiotics, as Sophie showed. But we believe that AI can be used not only in drug discovery, but also in other cases, such as diagnostic, biomarker design and assessment, multi-omic and other bioinformatic analyses, antimicrobial susceptibility testing, etc. So obviously the potential uses are myriad. But this is a lot of theory, but there is some basis in fact. This publication came out just under 2 weeks ago, where they designed a drug to treat pulmonary fibrosis. And this was taken from target discovery to preclinical candidate nomination in just about 18 months. And to prove that, and to further demonstrate the success of this approach, this has now been in two successful phase 1 clinical trials. So it gives credence to the fact that artificial intelligence and machine learning can indeed help. So finally, we'd like to invite anybody to ask any questions, and I'd just like to thank my fellow speakers, Sophie and Lucia, for helping with this presentation. Thank you.